And then how do I progress? Right there. Okay, great. Thank you, Mitch and Eric, and thank you to our 4 speakers for fabulous DataBlitz talks. As an audience member, I always love the DataBlitz format. You get so much information so efficiently, and your neurons are just firing, and I just wish the rest of my life could be that efficient. But I know from the perspective of the presenter how much work it is to get it down to 5 minutes, and in particular how you feel you're losing the nuances and the subtleties and the granularity. It can just be painful. So thank you to our 4 presenters for helping us to consume your science in a really terrific way. So I'm going to try to hit some of the high points that I see are common threads and themes to get everybody thinking about questions. I'll try to stay within our 15 minutes since I know this is the last thing between you and lunch. So I'm going to start with discussing Erica Forbes' talk. Now, I followed Erica's research for a number of years. There's a lot of substantive overlap in what we do, and what I've always admired about her program of research is the systematic fashion by which she addresses a cognitive neuroscience question carefully in adults and then downward extends it to adolescents in a very appropriate sort of developmental framework. And I think we see that here. So Erica has demonstrated very elegantly that adult depression is associated with hypofunction in the brain's reward circuitry and made linkages to EMA as well as linkages to anhedonia. And here we see this downward extension to adolescents with depression. And I think what's the really fabulous piece here is the ecologically valid and developmentally appropriate stimulus set. Those BFF videos are terrific. First thing I'm going to do Monday is email you for a copy of those videos. I think they're wonderful. And I think the appropriateness of those videos comes through in the subtlety of the results and the new findings relative to adults with depression. So there's some of the usual suspects, ventral striatum and other aspects of the reward circuitry, but now we're seeing this hidden medial prefrontal cortex, as was mentioned, which has linkages to self-referential processing, as well as precuneus, which in other contexts has been linked to rumination and depression. And so by using this developmentally appropriate stimulus, we have novel insights into endophenotypes relative to adolescent depression. So I think that was very appropriately done in a developmental context. I'm going to go out of order here. Just in my mind, it was easier to link the 2 fMRI talks together. So we also heard from Karen Rudolph, a wonderful study about relations between history of social rejection and depression risk in adolescent girls using the Cyberball task. And this is sort of my first common theme here, is the use of a developmentally appropriate task. What could be more relevant to adolescents and adolescents with depression than social exclusion, social development? And so this Cyberball task, I've talked to folks who've used this task, and for teenagers, this can actually be an upsetting task. I mean, they come out of the scanner and they need some debriefing. So I think it's a very appropriate task. Once again, we see novel insights linked to this task, this really cool, really interesting interactions in dorsal anterior cingulate with linkages to social pain, and in the amygdala with linkages to threat, only in those girls with a prior history of victimization. So a really well thought out and elegant analysis and elegant result. So in terms of a very brief interim summary and some common themes across the neuroimaging studies, I know I'm preaching to the converted here, but kid brains and adult brains are very different. And we've known this for 10 years or more from the analytics side. Our software packages keep trying to make it easy for us to analyze our adult data as though it were kid data, and that's not always the way to do it, and now they're pediatric templates. But these talks, I think, really emphasize the other side of that coin, is that you have to use developmentally appropriate stimuli. You can't just use what's worked well in adults to study kids. I keep saying kids for adolescents, and I realize I'm old enough and I'm able to do that. So when I say kids, I mean adolescents. And another theme is that even though outwardly Dr. Forbes' talk was studying positive valence systems or reward processes, and Dr. Rudolph's talk was more threat and negative valence systems, we see this social overlay to both tasks. And I think this just drives the point home that social systems and social processes are so key for adolescents that even when studying another construct, it's likely the case that the best way to do that is to incorporate a social aspect to the study or to the stimuli. And what I hope this might start to signal is perhaps acceptance for the beginning of development of a pediatric or adolescent cognitive neuroscience toolbox. I know that in adult research. There's been traction for toolboxes. Deanna Barch's group in the Matrix Consortium studying schizophrenia has really pushed rather well, I think, the use of specific tools to study social deficits in schizophrenia. And I am a big believer in, if in order for big data to work, in order for us to compare data across labs, we've all got to start using similar experiments. We can also do our pet studies on the side, but only through the use of a toolbox will we truly be able to link data, in the fashion that was described earlier in terms of uploading our RDoc data to the RDoc data repository. And in terms of RDoc itself, I was driving over here this morning and trying to think like a developmentalist, and I was trying to think, what developmental stage is RDoc in? And I was trying to think of the conversion of human years to RDoc years, and it seemed to me that we've been talking about RDoc maybe for 3 or 4 years, so maybe I have a 10-year-old boy at home. It sounds about right. It's like, he's like a pre-adolescent. I know he's a good kid, but I don't quite know what he's going to be like in the future yet. And I feel like that's where we are with RDoc. It's still experiencing some growing pains. I hope I'm not offending anybody when I say that. And in particular, with relation to adolescent research, the 5 constructs that NIMH has delineated for RDoc, positive and negative systems, social systems, working memory, arousal, that might not be the best way to cut nature at its joints for adolescent research. And I think one example of that we saw in these 2 talks is that social systems might be so primary in adolescence that even if you're studying positive systems, as Dr. Forbes was, the social piece is going to be very important. And even if you're studying negative systems and threat systems, there's going to be that social overlay. So the social RDoc construct might be more of a cross-cutting construct than a separable one as it is in the adult matrix. So we learned about stuff that Dr. Gibb thinks is neat, and now we all think it's neat also. Some wonderful multimodal research incorporating reaction times, electrophysiology, and eye tracking to predict depressive symptoms in kids. I was particularly interested in the pupil dilation work, the ability of pupil reactivity to predict depression symptoms 2 or 3 years out is just incredible. And for my money, of all the translational clinical neuroscience tools we have at our disposal, eye tracking and pupillometry to me seem like they have the greatest chance of winding up in a pediatrician's office in the not too distant future. And some groups are even experimenting with that. I know one group at UCSD doing that now. Calibration is simple, it's non-invasive, there are algorithms just to score it up. And if that's the case, and if pupillometry can predict the onset of depressive symptoms in 2 years, sign me up. I mean, I think that is going to be a real clinical deliverable in the not too distant future. Wonderful work on conditioning models for the acquisition of biases, looking at the late positive potential, this slow forming midline ERP component sensitive to emotional valence, and that really fabulous neural synchrony data. We spend so much time thinking about the transmission of risk for depression from parent to child, and there are obviously so many factors that play a role there. But if it's the case that when parents and their children interact, their frontal brain asymmetry becomes synchronized, that's a very powerful addition to our knowledge about how that intertransmissional risk occurs. And then I was pleased to see the cross-species studies looking at interleukin-33 in the rat and reaction to stress, and certain haplotypes in humans and their response to severe stressors as well. And then last but not least, Dr. Benjamin Henkin told us about molecular genetic influences, specifically the 5-HTT-LPR, the serotonin receptor gene, and subtle interactions with negative reactivity, stress, and attention in youth, and the development of depression and anxiety 3 years later. Just some really exquisite, again, multimodal research. So in terms of common themes for these 2 talks, and then I'll try to put a bow on it on the next slide, we see the richness of multimodal data assessment, that these constructs that we're talking about cannot just be recorded through self-report, cannot just be understood in a scanner. None of these is the truth, none of these is the gold standard. As a neuroimager, I often get asked, so what's really going on? And it's just one piece of the puzzle, and you need to measure all these variables to really understand a construct from all angles. And this notion that what is the right press in the laboratory to elicit a response response from the child that is relevant to your research question? Is it a structured interview? Is it doing a computer task? I just love this notion that child interacting with mom may be one of the most potent or sensitive presses that we have at our disposal. And the daunting challenge, which we've heard about today, of incorporating molecular genetics. Every molecular genetics talk I go to, I learn that everything we knew 6 months ago maybe no longer is correct now. So it's a tough field to keep up with, and kudos for efforts to nevertheless dig our heels in and try to make sense of those interaction effects. So my sort of thoughts and future directions, and maybe to get you all percolating for some questions to our speakers. As I've alluded to, studying adolescent depression or any kind of developmental psychopathology research is more than just studying kids. That we have to put this work in a developmental science context and developmental science framework and understand trajectories of development, understand moderators that put some kids on a path to resilience and others on a path to risk. In this funding climate, it's daunting to think about the resources it would take to, say, scan 100 kids every year for 10 years, but maybe through collaborations, this is really the kind of work that needs to be done to understand those trajectories of risk. I hit on the importance of multimodal measurement, but I think all too often, and I know I'm guilty of this as well, we collect all this data and then we opt for the sort of striptease approach to publication where you publish the reaction time data and then you publish the EEG data, and it seems to work, and those of us who have recently gone through a 10-year review process, there's a logic to that approach. But I think the real richness in those data is to analyze them together, because each piece of information is telling you something unique, whether you form latent factors and do structural equation modeling or take another approach. If it's possible to keep the richness of those data intact by incorporating multiple units of measurement, I think that's the way to go. We're always faced with the translational divide, and I think we've all heard the criticism that as scientists we study questions rather than provide answers, and this is a criticism that keeps me up at night. I mean, I really think we can't stop until we have deliverables that can improve, decrease suffering in children with depression. I want to make a push for integrating our work with our preclinical colleagues. We saw some of that today. I think the advances in tools available to preclinical neuroscientists, even in the past 3 to 5 years, are just breathtaking. The advent of optogenetics to teach us about neurotransmitter systems and specific cell types and animal models, and clarity from Carl Disseroth that provide this view into a living brain. It would really, we're losing an opportunity if we don't make an effort to reach out to our preclinical colleagues. Some final thoughts on RDoC and DSM. We've all aired some of our thoughts earlier today. I think we all like it as a concept, but are still figuring out how to implement it. And I think we're at a funny time now where we're trying to do things both ways. And I don't know if that brings us any closer to decreasing heterogeneity, which I think is the ultimate goal of RDoC. So I think some of us are going to have to decide to go all RDoC, or to decide that maybe another approach for our particular research question is correct. But I think trying to do both at the same time is not necessarily what any of us had in mind. And then my final thought is, with all this work that we do trying to predict depression risk in youth, the ultimate question always is, how young can we go? If an ounce of prevention is worth a pound of cure, the earlier we can identify risk markers, the better. I know in the autism field there are prospective risk markers in children as young as 6 months of age using DTI and using eye tracking, and only when we really push the envelope in terms of how young can we go can we really prevent the onset of these terrible disorders, which is the reason why we all do what we do. So thank you, and we'll open up for questions. Can I go sit down? Oh, yeah.